And I must say, it was an unexpected honor when I was asked to come and give this inaugural lecture or opening lecture, because as you heard, if a biophysicist is, caught between physics and biology, we're caught more between biophysics and biology. So we're, a little closer to biology than most biophysicists, I think. And therefore, it's particularly nice to be able to come here and speak to you today. So Helmut raised this issue of this tension between doing physics while respecting biology, or sort of glancing at biology. And one other way to phrase that is in terms of the Scylla of physics and the Charybdis of biology, and that's where we all are in one way or another, right? This is from the late 18th century, actually. This is Prime Minister Pitt in the UK, or England in those days, steering the Britannia ship safely between, if you read carefully, the rock of democracy and the whirlpool of arbitrary power. So clearly he set his aim for something that's not complete democracy and not complete arbitrary power as well. And I guess that's what eventually developed into England and the UK. A contemporary version of the same myth. I don't think I need to explain this one to you. But of course, as scientists, we all have, I think, our personal Scyllas and Charybdises. I don't know, Tony, is Charybdises the plural of Charybdis? Yeah, he says yes. So our own personal Scyllas and Charybdises. And of course, in biophysics, the main ones are physics on the one hand with its precision and its theoretical underpinnings, and biology on the other hand with its complexity and in one sense lack of theoretical underpinnings. And that's where we have to navigate between these two attractors, if you will. So in our case, we have trying to navigate these waters by asking, we think, physical questions, or framing our questions in terms of physical concepts, but using mostly the methods of biology to address those questions. And that's been a very interesting challenge, and it's still, it's very engaging to us to try to think in those terms. And in particular, we have been interested in how membrane proteins are made in the cell and how they get inserted into the membrane, and how we can understand the underlying physics of this rather complex biological process. So of course, biophysics has studied protein folding, both soluble proteins and membrane proteins, for a very long time. But there has been one aspect of folding that has been very difficult to address using sort of standard biophysical methods, and that is the co-translational aspect of folding that we see in cells. It's fine and well to study folding of purified proteins, unfold them, refold them, and so on, but we still don't really know how far that knowledge explains what's really happening in the cell, where a lot of these processes are co-translational. So typically, membrane proteins, nearly every membrane protein is inserted into the membrane co-translationally and even start to fold co-translationally. And so there's one issue is how does that affect the final structure? Is there kinetic determination and so on? Secreted proteins are also, of course, secreted across the membrane co-translationally, so that's a process that is intimately involved with protein synthesis and ribosomes and translocons. That is difficult to model in a pure biophysical system. And finally, of course, soluble cytoplasmic proteins, many of them at least, initiate folding co-translationally, and some fold almost to completion co-translationally, as I will show you later on. So how do we address, how do we come to grips with co-translational processes and trying to think about the physics of the underlying physics? That's what I will talk about. So I'll spend most of my time on membrane proteins and just get in a little bit about soluble proteins at the end. So let me just summarize what the structures of membrane proteins have told us. And that, interestingly enough, if you don't worry about the specific functions of membrane proteins, but just about their overall structural characteristics, they can pretty much be summarized on this one slide, what we have learned from the structure so far. Membrane proteins are composed of transmembrane alpha helices. These helices are hydrophobic to interface with the membrane, obviously. They often end or have tryptophans and tyrosines, aromatic residues near the ends. I think I need a better. Tryptophans and tyrosines tend to be near the ends of these helices to anchor somehow the protein to the interface of the membrane. And finally, you have this very highly biased distribution of positively charged residues when you compare the inside of the inside facing parts of the protein, the cytoplasmic facing parts, to the outside facing parts of the protein, so-called positive inside rule. And this pretty much describes in a general sense the structures of the alpha helical membrane proteins. Now incidentally, I just realized when I was preparing this that we were hearing about anniversaries here, you know, what was it, 30 years anniversary of the European Biophysical Society, and this is the 10th the 10th meeting and so on. And I realized that actually much of this basic concept also has an anniversary this year. So it's actually precisely 40 years ago that the first sequence of a transmembrane segment was published. And that was from glycophorin A. And you see it was immediately obvious that there was this long stretch of hydrophobic amino acids that it was easy enough to postulate that that spans the membrane. And if you just look at the length here, about 20 residues, and the known thickness of the membrane, you could immediately deduce that this is forming an alpha helix, right? So that was 40 years ago. Actually, 1975 was a good year for membrane proteins because the same year, the famous Henderson and Unwin structure of bacteriorhodopsin was published. And that's when we realized that helical membrane proteins form bundles of alpha helices, are the basic architectural principle of these proteins. So we've actually known this for 40 years, but we still don't understand fully how these things are made. Actually, in my own case, I realized that it's almost an anniversary as well, not quite, but 29 years ago is when we published this paper on the bias of positively charged residues between the two sides of the membrane, the positive inside rule. So that entire slide that I showed you has really been around for 30 years now, and we're still trying to work out the implications of it. So let's now move to the co-translational aspect of membrane protein assembly. So this is a much more modern view of the system. This is only from last year. It's a cryo-electron microscopy structure of a mammalian ribosome bound to the so-called SecM translocon in the endoplasmic reticular membrane. And it's through this translocon that all secretory proteins are translocated across the membrane, and all membrane proteins are inserted into the membrane. So for membrane protein assembly, the idea is that if you have one of these long hydrophobic stretches that will eventually form a transmembrane helix, it will, of course, be polymerized in the ribosome. It will be pushed down the ribosomal tunnel. It will come into the SecM translocon. And at this point, if it has the right properties, it will be shunted sideways into the membrane to form a transmembrane alpha helix, rather than just passing through the translocon to become part of a secretory protein. So this is the heart of the biosynthesis of transmembrane alpha helices, to understand this process of how do they trigger this, how is this triggered, this exit from the translocon into the membrane. Now, from a physics point of view, of course, one thing you really want to know here at the outset is what are the energetics of this process? You know, what are the energies involved here? Just how many kcals per mole of insertion free energy does a transmembrane segment have when it comes through the translocon going into the membrane? Where's the threshold? How many hydrophobic residues do you need in a segment for it to become transmembrane? So this is the first story I'll tell you about. This was a problem that we confronted about 10 years ago. And of course, the question is then, how do you measure this? You can measure peptides partitioning into membranes, but how do you measure partitioning of different hydrophobic segments being made on the ribosome, co-translationally partitioning into the membrane? How do you measure the energetics? And this is something that became a fantastic collaboration with Steve White, who is in the front row here, that we spent a number of years trying to work this out. And the basic idea we had was that it's probably very difficult to find a pure biophysical method to measure these energetics. So let's see if we can come up with something, borrow some methodology from biology, from cell biology or molecular biology that can allow us to do these types of measurements and get quantitative data. So the assay we came up with was to use a very well-established system for in vitro synthesis of membrane proteins. So you just take a plasmid encoding your protein, you transcribe the gene into mRNA, and you put that mRNA into an in vitro translation system, just a lysate or in vitro translation system that can translate this into protein. There are many such systems established. And now in order to make it insert into a membrane, you have to add membranes to that in vitro system. The membranes you add are called rough microsomal membranes. So these are derived from the endoplasmic reticulum of mammalian cells, in this case dog pancreatic cells, from which you can isolate ER-derived membrane vesicles that actually retain the propensity to insert transmembrane helices and make membrane proteins. So we figured maybe we could use this methodology. Of course, we need to somehow figure out whether a particular segment in that protein we make is a transmembrane helix or not. And so we took advantage of a very nice property of these rough microsomes, namely they contain an enzyme that can add what's called so-called glycans, that is sugar moieties, to specific residues in the nascent chains, specifically asparagine residues, if they're in this little tripeptide acceptor context. So if you have one of these NXT sites in your protein, and if that NXT site gets across the membrane into the lumen of the rough microsomes, the enzyme will add a glycan, which increases the molecular weight of the protein sufficiently that you can see a shift on a gel, whether it's received the glycan or not. So the enzyme is only present on the inside and not on the outside. So if a specific site is glycosylated, you know it's on the inside. So that's what makes a very simple experiment possible, namely to take a membrane protein, and this is the one we happen to use, which has a large domain that is normally translocated across the membrane into the lumen of these vesicles, introduce any kind of hydrophobic segment in that by genetic engineering into that protein. This is a typical segment that we use, very simple amino acid composition with leucines and alanines, and then engineer on either side of this one glycan acceptor site. So the idea being that if your hydrophobic domain has the right properties to partition into the membrane during translocation, only one of the two sites will make it across the membrane and receive the glycan. Whereas if you have a segment that doesn't get inserted into the membrane, it will all be translocated and both of these sites will end up being glycosylated. So there will be a molecular weight difference between these two forms of the protein. So here is a typical gel where this is in the absence of such rough microsomes, but in the presence of membranes, you can see that this particular transmembrane domain engineered into this construct is mostly singly glycosylated. So this band represents molecules that received a single glycan, and the upper band correspond to molecules that have received two glycans. And you see this particular one is mostly singly glycosylated. So this is a good transmembrane segment. It inserts pretty efficiently. On the other hand, if you now change this segment, in this particular case, we just change one amino acid from cysteine to proline, now you see that all the molecules essentially receive two glycans. So this is not a good transmembrane segment. It just passes through the translocon into the lumen. So I think you can now see quite quickly how by making lots and lots of different segments here, where, for instance, we move a proline from one end to the other, or we move a cysteine, or we take any of the other 20 amino acids and we move it successively through the segment. So we make a lot of these constructs, and for each one we measure basically the partitioning equilibrium between these two forms of the protein by quantitating these bands. We should, at the end of the day, end up with free energy estimates for how much any given type of amino acid contributes to membrane insertion at a given position in the sequence. And to make a long story short, this is what you see on this slide. So here is, a couple of hundred constructs that were analyzed in this way, and from all the data we teased out the individual contributions of each amino acid as we moved it across the hydrophobic segment. So what you can see here is the contribution to the overall free energy of insertion from an alanine, for instance, in positions from the N-terminus to the C-terminus to the segment. And for alanine, it's pretty much zero, no matter where you put it. That means alanine doesn't really care. It's equally happy in the membrane and outside the membrane. But if you take something like leucine, you see it's all negative, the blue curve. So it promotes insertion wherever it is. If you take something like aspartic acid, it's always positive, so it always works against insertion. And you can see exactly by how many kcals per mole. And you can also see that if you place this charged amino acid right in the middle of the hydrophobic stretch, it's a lot worse than if you put it at the ends. So here we have a really rather complete picture of how each of the different amino acids quantitatively contribute to the insertion. So we have, so this is the energetics of membrane insertion. You can work this into a prediction scheme where you just sum up the values for the individual amino acids in the segment, and that turns out to predict the insertion efficiency, the insertion free energy quite well. One can actually take this system a little bit further, and this I thought I was particularly pleased with these results. So again, old techniques have been developed, to introduce non-natural amino acids into these stretches, into any in vitro synthesized protein, in fact. And one method that's very easy to use is to put a stop codon into the position where you want your non-natural amino acid, and then you program your in vitro translation system, you put in an extra tRNA carrying your non-natural amino acid, and that tRNA is a so-called suppressor tRNA, which means that it can read stop codons. So you basically have an extra amino acid. You have 21 amino acids in your genetic code by doing it that way. So now you can program, you can directly determine where you want your non-natural amino acid introduced, and by modifying the tRNA with the particular amino acid you're interested in, you now have access to pretty much any amino acid you can imagine, and you can test it out in your system. So it turns out that a very good way to put the amino acid onto the tRNA is to use something called flexozyme that was developed by Hiroaki Suga. It's a ribozyme that can put tRNAs, no matter what the side chain is, onto the suppressor tRNA, and then you can use it in the in vitro reaction. So one example of how you can use this to get an even more detailed understanding of the energetics of insertion of membrane proteins is demonstrated here. So here we took a series of non-natural amino acids that only differ by a single methyl group. So one methyl group, two methyl groups, three methyl groups, just a long linear alkyl chain, going from one methyl up to 8 methyls. So just string on the methyls one by one, and put them in this middle position, and measure the insertion energy, free energy for each of these constructs. And that actually gave us quite nice data. So the red dots here are for these increasingly long side chains, alkyl side chains. And here you see the drop in free energy of insertion as a function of side chain length, or actually as a function of accessible surface area. And if you look at the slope of this, it works out to actually 10 calories per mole and square angstrom. So that's the hydrophobic effect, if you will, in this in vivo, complex in vivo insertion system. We also did the same, but using aromatic side chains up to biphenyl is the biggest one here. And now if you work out the slope, it's actually 7 cals per mole and square angstrom. So this was, I don't think I had expected when we started this, that we would be able to use this kind of very gross biological experimental setup and actually get rather precise values for the hydrophobic effect in this complex system. Now, it's quite another matter is to interpret this value. What does it really mean? And that has spawned a lot of work and a lot of discussion downstream of this work, so that, which is still going on actually, so that's quite nice. Okay, so this was for the endoplasmic reticulum. Of course, it's nice if you can do this for other types of membranes as well, and I'll just show you one example, and that is for E. coli. So it would be nice to do the same type of experiment, but for insertion into the E. coli in a membrane rather than the endoplasmic reticulum in mammalian cells. In E. coli, you cannot use glycosylation. They don't have glycosylation machinery. So we came up with a different scheme to do the same type of experiment, which was actually based on a spurious observation that Kuriaki Ito in Japan made when he was working with a protein called lactose permease. So it's a lactose transporter in E. coli, LAC-Y, lactose permease. And he discovered quite serendipitously that if you express the second transmembrane domain of this 12 transmembrane segment protein by itself and allow it to integrate into the membrane, it will actually become a substrate for an intermembrane protease called GLP-G. So it will be clipped if it becomes transmembrane. So we designed this system, which is essentially the same as the previous one with these test segments that we can vary the hydrophobicity of. And now we just fuse this LAC-Y TM2 downstream. So if the hydrophobic segment inserts in the membrane, in the inner membrane of E. coli, this is left outside the membrane. It's not clipped by the protease, whereas if this one does not insert, it gets translocated across the membrane. Now the protease clips it. So again, we can read the readout from the assay is just 2 bands on a gel, the full-length band and the clipped band, and we can quantitate how much we have of each. And then we can repeat the same analysis as before. So doing that, we could, again, quantitate the contribution of all of the different amino acids to insertion, free energy. This is now for the middle position in the transmembrane segment. And here they are. And it turned out that they actually correlated, I would say, surprisingly well to our endoplasmic reticulum results. So here is, for each of the 20 amino acids, the delta G contribution in the ER system versus the E. coli system. And you see it's not a bad correlation. And actually the slope here is 1 actually. So for those two systems behave very similarly. Maybe not so surprisingly because the two insertion machineries are very similar, but of course the lipid composition is quite different, so you would not necessarily have expected this. Okay, so Translocon-mediated insertion. We feel we have a decent understanding or a decent picture of the energetics of this, quantitative picture. As I said, it's still discussed exactly what determines these numbers, but at least we understand the energetics on some level. So the next question we asked ourselves is, okay, so we have kind of an equilibrium picture of what's going on, but what about the actual process, the kinetics of the process? Is there any way we could, in some easy way, gain access to follow, find a method where we could follow the progression of a transmembrane segment down the ribosome into the translocon and into the membrane, and somehow see how, as a function of chain length, where are the critical steps in this process? And one thought we had was that, well, if you have a transmembrane segment, if you imagine it coming down, this is Gedanken experiment, if you imagine it coming down the ribosome and just starting to touch the translocon or possibly the membrane, if this is very hydrophobic, there is a large energy gradient for inserting it into the membrane. So if it starts to make contact, that would be a downhill process for essentially pulling it into the membrane. If that happens, it would stretch the nascent chain. That would increase the tension in the nascent chain. So if we could somehow detect that increase in tension, then maybe we could follow this process and see at what chain length it happens and so on. So how do you follow tension? How do you detect tension in the nascent polypeptide chain inside the ribosome? That was our question, which again, thinking in terms of pure biophysics would probably be very complicated. At least we could not come up with a good idea of doing that. But again, biology helped us and gave us an opening that we could use. So what I will now switch to is to tell you about how we measure indirectly, for sure, but still in some sense measure forces acting on the nascent chain, inducing tension in the nascent chain. And this actually, the solution came from kind of an unexpected biological discovery, which was something called translational arrest peptides that you may have heard about. So the first one was, or one of the first ones was discovered by Koryaki Ito in Japan. And what he found was that there's a protein in E. coli called SecM. And when the ribosome translates, it's a secretory protein, so it has a signal peptide. And when the ribosome translates this SecM protein, it tends to stall before it hits the stop codon. It tends to stall on a short piece of peptide that he called an arrest peptide. So the ribosome tends to stall quite efficiently on the last codon of this little piece, little peptide that he called the arrest peptide. So that was interesting, and the cell is using this for regulatory purposes. So that was the biological interest. But what caught our attention was that early on there was a speculation or a suggestion saying that one way for the ribosome or for the system to overcome this stall, translational stall or arrest, and continue translation to the end of the message, is if there is tension in the nascent chain, exactly at the point when the ribosome hits that final codon in the arrest peptide. If that's the case, then the speculation was, then the ribosome will no longer arrest, but it will continue translating beyond the arrest peptide and finish up making the entire protein. So that's what caught our attention. If that's true, then we should be able to take this very tiny little piece of the protein and use that as a force sensor and transplant it into any other protein context that we'd be interested in. So we'd essentially have an internal force sensor that we could engineer into a protein, and that, of course, we could put that into a nascent chain, and that would allow us perhaps to sense or to detect tension in the nascent chain as a function of or during elongation of the protein. So that's what we set out to try. We used a couple of different arrest peptides that people had identified. This is the original one from the E. coli SecM protein. You see it's fairly long, 17 amino acids, and some of them are more important than others. And in particular, its proline at the very end is super important. So if you mutate that, you get no stalling at all. But we also used two shorter ones from another bacterium. And it was shown by other people that this is not a particularly strong one. This is a fairly strong one. And this mutant that we made in this short one, turning this isoleucine into a proline, is even stronger in the sense that you need to pull harder on the protein in order to overcome the arrest of that segment. So those are the tools we have now to try to follow or to detect changes in tension in the nascent chain. So I'll show you two applications of this. The first one is for membrane proteins, just the problem I mentioned before, is that, can we detect interactions between a hydrophobic segment and either the translocon or the membrane using this technique. So the idea is to make a series of constructs which are very similar to the ones we used to measure energetics. You may recognize the outline of the protein. Here is our hydrophobic segment that we can make any sequence we want. Here is the arrest peptide. And the only thing we vary here is the separation between this hydrophobic segment and the arrest peptide. So we make a series of constructs where we place the arrest peptide anything from very close to the hydrophobic region to further and further away. So the idea would be that if it's very short here, they're very close together, the hydrophobic segment will still be in the ribosome when you get to the arrest peptide. No reason to think that there should be a particularly strong pulling force, so we would expect efficient stalling at that kind of construct. Going to the other extreme, where we have a very long separation, we would expect the hydrophobic region to be already integrated into the membrane when the ribosome gets to the arrest peptide. And hence, again, there's no real reason to think that there should be a strong pulling force if our ideas are correct. But somewhere in the middle, maybe we'll see something. that could reflect interactions here between the hydrophobic region and the insertion machinery. So we made all the constructs, we expressed them in vivo actually, so you can do this experiment in vivo now. And we just looked at, for each construct, for each length here, what fraction of the molecules that we made in a short 2-minute pulse radioactive pulse were arrested versus full length. And the more full length, the higher the pulling force, right? The more full length, the higher the tension in the nascent chain. So essentially we can construct a force profile as a function of length of the intervening sequence. So what we found was this. Well, first I'll just show you the underlying experimental data, which is very simple. It's just a gel with 2 bands on it. So this is the whole experiment really. So here is one example where we have a very long separation between these two. This is after we label the protein for 2 minutes and immune precipitate it out of E. coli and run it on a gel. This is the arrested form of the protein. This faint band is the full-length form of the protein. And we know that because if we mutate that last proline in the arrest peptide, it no longer works and we only get the full-length protein. So we just get molecules ending here. But you see that with this long separation, the arrest peptide arrests very efficiently and we get almost only arrested protein. However, if we now vary L, the separation, and just quantitate the amount of full-length protein, the fraction full-length protein, as a proxy for the tension in the nascent chain, it's not piconewtons, it's fraction full-length protein, but there are ways to convert between the two. This is what we got. So here you have length, fraction full length, very short, no full length, very long, no full length. In between, not 1 but 2 peaks actually. And so we spent some time trying to figure out what they mean. We think that the first one, because we can show by mutagenesis that it only involves the most N-terminal part of this hydrophobic region. So we think this is an early interaction between the hydrophobic region and possibly the translocon itself when it comes in, just comes out of the ribosome. But the main peak, if you look at the length of the separation here, compare it to the known dimensions of the ribosome, and also it's because it's sensitive to mutation throughout this hydrophobic region. We think this actually represents the insertion into the membrane. This is when the hydrophobic region is exiting the translocon into the lipid bilayer. And so this happens at about 40 or so residue separation between here and here. Just to show you that it's sometimes good to have more than one type of arrest peptide, we redid these experiments with a weak one, the E. coli one. This was a medium strong one, I should have said. The E. coli one is very weak, so we don't need much tension to actually get complete full length. And so we lose all this fine structure, as you see. We don't really see these 2 peaks anymore. We just see one very broad peak because it's so sensitive in this particular system. The forces are too high, essentially, for the E. coli, for the E. coli arrest peptide. We have other systems where the E. coli one is the perfect one in terms of reacting to the force levels we're interested in. But we also took, more interestingly, the mutant arrest peptide, that's the stronger arrest peptide, and redid the analysis, and this is what it looks like now. So here is with the strong arrest peptide. You see the first peak, whatever force is generated here is not enough to overcome the stalling on this more potent arrest peptide. And even this peak now sharpens up. This main peak sharpens up considerably, and it's only a couple of residues across now. So we can really pinpoint very precisely where the maximum tension in the nascent chain is generated in terms of chain length. So this shows you that we can fine-tune the arrest peptide for the problem at hand, basically. So the conclusion from here is that we have during membrane insertion, there's actually an early interaction and a late interaction. In both cases, there is some extra force, some tension generated on the nascent chain, but and those are the 2 stages we think we're seeing there. So finally, the last example I will give you is when we're now moving to a cytoplasmic protein instead. So this is now co-translational folding of a cytoplasmic protein, again using the same arrest peptide setup. So the idea here, of course, is very similar to before. Assuming that we have some domain that can fold co-translationally at some point when either the tunnel becomes wide enough or when it's just outside the tunnel, so there is space enough for it to fold. You can imagine that if the tether is too short, if the distance between the arrest peptide and this domain is very small, there is not enough room in the ribosome for it to fold, and hence it should not exert any force on the nascent chain. Likewise, if you make a very long separation between the arrest peptide and this domain, it should be well folded long before the ribosome gets to the arrest peptide, and again, there should not be any particular pulling force generated. On the other, but we can imagine an intermediate situation where the protein begins to fold at about the same time as the ribosome gets to the arrest peptide. And so if you're in this confined environment of the ribosome, you can imagine that you have the chain length is just long enough that if you pull it straight, the protein can fold. So that will increase the tension in the nascent chain, right, for that particular chain length. If you make it longer, maybe it will be reduced, and finally it will go away again. So maybe we could pick up that kind of folding force using the arrest peptide as a sensor. And here we now went to the E. coli arrest peptide because it was weaker and we didn't know what to expect, so we thought we'll just take a very weak one and see if we can see anything. The model we chose was the tiniest domain we could find that had a nice two-state folding and fairly stable folding. So this was for the kind of the pilot experiment. It's a zinc finger domain called ADRI1. ADR1A. It's only 29 residues long. Here is the sequence. It has 2 cysteines, 2 histidines that coordinate the zinc, so folding is completely dependent on zinc. If there's no zinc, it won't fold. If there is zinc, it folds quite efficiently. So we like that because it also meant that we could control the folding state by controlling the zinc levels during translation. So we just made a simple series of constructs with this ADR1 domain, different separations to the arrest peptide. And then because this is so small and we still wanted to look at this on gels, we just added some unrelated sequence that doesn't fold in itself on the N-terminus to make the whole protein big enough that it would be easy to resolve on a gel. And then we just, like before, we translated all these constructs. Actually, here we translated in vitro rather than in vivo, at least most of the experiments, because it was easier to control the zinc levels in vitro. So we used an in vitro translation system. And here is what we found. So if we now translate this series of constructs that vary by in this separation L, in the absence of zinc, actually in the presence of a chelator, to make sure there's no zinc, free zinc in the translation buffer, and we used a commercial in vitro translation system called the Pure system. We don't see anything. It's all very well arrested, no matter what the separation is. However, if we include zinc now in the buffer, in the translation buffer, and repeat the experiment, we see something very different. So in the presence of zinc, we now see a strong tension being generated in the nascent chain with a maximum at a separation of about 25 residues from here to here. So the total tether length is 25 here. Now 25 residues is not long enough to allow this zinc finger domain to exit the ribosome tunnel. So it seems like it's folding inside the tunnel. That's what we think. We also repeated this experiment in vivo just to see if we could see anything. And it turns out we do see a small signal in vivo as well. The problem is to load up living E. coli cells with enough zinc to see much. So we had very high levels of zinc in the medium, but how much really got into the cell and wasn't pumped out again, we don't really know. But apparently it was enough that we could see a small signal, at least in vivo as well. So it appears that we can somehow follow folding also in vivo. And finally, just to see if it was really feasible for this thing to fold in the ribosome, we thought maybe we can be able to visualize this folded domain inside the ribosome if we're really lucky. So what we did then was we made essentially the same construct. Here is at the top of the top of the peak of the force profile, 25 residues, that's where we have the maximum pulling force. So we were interested to see where is the protein at that pulling force. Of course, you know, with the E. coli arrest peptide at 25 residues length separation, we have only full-length protein. We have no stalled protein. So there's no way we could purify ribosome nascent chain complexes with this folded protein in the ribosome. So what we did was we took one of these we took this very strong arrest peptide instead. We replaced the weak E. coli arrest peptide by this much stronger mutant arrest peptide from the other bacterium. And this now stalls it, even though the pulling force is quite high. So here we could, with the help of Roland Beckmann in Munich, we could actually prepare ribosome nascent chain complexes using this construct with a purification tag on the N-terminus, so you can pull down the ribosome nascent chain complexes that way. And he put it in his cryo-EM setup, and actually to our, I would say, delighted surprise, he came back with this structure for us. So here is the nascent chain going down the ribosome tunnel, exit tunnel, and here is now a new density that they had never seen in their ribosome structures before with other types of nascent chains in it. It's the right size to correspond to the folded zinc finger. We know the structure from NMR of this one. And so, and it's, you know, it's fairly deep in the tunnel. It's about 60 angstroms away from the P site and maybe 20 angstroms or so from the entry into the exit tunnel, or the exit rather from the tunnel. And actually you can maybe appreciate the fit better on this slide. It's not super high resolution given what they can get in the best cases now, but 4.8 angstroms is still enough that it was possible to model the NMR structure into this density, and you can see the fit is quite nice. So it appears that we can not only you know, detect the folding of this little domain by the force measurement assay, but we can also, by increasing the strength of the arrest peptide, we can trap it in the ribosome, and actually in this case, maybe we were very lucky, but in this case we could actually see it now by structural means inside the ribosome tunnel. So at least these very tiny domains can fold inside the ribosome, and of course now the question is, you know, how much bigger can you make it and still find, or how much bigger a domain can you put in there and still see it fold inside the ribosome, and what happens if you make it really big, and you know, how far out does it fold, and so on. So we have some work to do. So that's basically where I want to end. I hope I've shown you that at least in certain instances one can adapt methods from cell biology, molecular cell biology, make them quantitative, and address questions that are somehow, at least conceptually, physics questions, and give us some insights into certain processes, in this case, co-translational processes. You know, speaking in terms of Scylla and Charybdis, I think, to come back to this, we are continually trapped between these two attractors, between the, let's say, the promise of very well solid, intellectually rigorous science on the physics side, and the perhaps temptation to embed yourself in the great complexity of biology without perhaps understanding all the, you know, the deep details of what you're doing. And I think we all have to navigate this daily. You know, where do we want to be on this scale between biology and physics? And maybe we should try not to get too close to either one, but I think it's a matter of personal taste, which side of the strait between the Messina Strait that we want to rather be at. So I think that's all I wanted to say. I won't go through all the people that have contributed to this work, but I'll just provide a greeting from them, and hope you will have a great, or we will have a great conference in the coming days. Thank you.